Hello everyone. Today I'm going to talk about my experimental design. I want to study about microglia in Alzheimer's disease. As we all know that ApoE plays a crucial role in central nervous system as it's involved in lipids metabolism with the ability to bind to lipids and transport these lipids to neurons. Additionally, ApoE assists in clearing amyloid beta by taking up the proteins before they can accumulate and form larger clumps that may clog blood vessels. ApoE also help regulate homeostasis in central nervous system by releasing signals that prevent apoptosis, promote synaptic formation, and stabilize calcium gradient. Research also show that ApoE co-localize with amyloid beta levels in the brains of patients with the disease. Amyloid beta is a byproduct of a protein called amyloid precursor protein. When this amyloid beta proteins accumulates in the brain, it can damage the neurons and synapses, ultimately leading to neuronal loss. Moreover, the presence of amyloid beta also contribute to the formation of neurofibrillary tangles, which also accumulate and then cause neuronal death. Furthermore, ApoE also involved in regulating neuroinflammation and linked to the microglial neurotoxicity, which can result in the loss of neurons and the synaptic network. When microglia engaged in repeated phagocytosis, they remain in an active state for extended periods, leading to an overreactive response towards the aggregates and neural cells. For my projects, I have two specific aims. The first one is I want to research on how ApoE isoform differently affect Alzheimer's disease, as ApoE has multiple isoforms that affect the progressions of the disease. I want to understand the underlying mechanism of this. As we all know that ApoE3 is the most common isoforms of ApoE, and it's associated with the neutral risk for late onset Alzheimer's disease, while ApoE4 linked to an early onset Alzheimer's disease. And this ApoE4 also characterized by severe symptoms and complex pathology of the disease. These two isoforms only different at the nucleotide at 112 position, which leads the ApoE3 to have a cysteine in its protein, while ApoE4 has an arginine. A recently discovered isoform of ApoE, which is the ApoE Christchurch mutation. ApoE Christchurch R136 S was discovered as a female from a family with an early onset disease history. She carried two alleles of this isoform. Therefore, she remained cognitively healthy until her 70s. And noticeably, although she has a lot of amyloid plaques accumulated in her brain, she never or barely formed any tau later in her life. And she has very healthy, less reduced in cognitive decline. I want to create a model line of this ApoE CRISPR by using CRISPR editing to introduce the gene into the mouse embryos. The target of this editing is that the ApoE gene will have one nucleotide change at the 136th position, so that ApoE CRISPR will form a serine amino acid instead of an arginine. This variance also associate with better gene phenotype for uptaking amyloid beta and delay in tau progression, and then minimize cognitive declines. The second aim that I have is that I want to study microglial functions in the Alzheimer's disease condition by differentiating microglia. I will differentiate microglia from iPSC, and I will use qPCR and IFA to confirm gene expression associated with each state of development during the whole differentiation protocol. And after I receive a mature microglia, I will test for the phagocytosis function of this cell type in vitro through the phagocytosis assay, in which microglia will migrate and surround the plaques areas. And I also consider to compare the active and inactive form of microglia through IFA. Furthermore, the derived microglia can be transplanted into the disease brain models to compare and contrast the functional and morphological changes of microglia in vivo. And as I expected, the microglia in the brain with ApoE CRISPR will show improvements such as less damage to the brain, be effectively clear up plaques, and it's also will slow down the tangles progression, as well as it will maintain its normal morphology. On the other hand, the microglia in the brain with ApoE4, which is the highest risk for Alzheimer's disease, will have dysfunctions such as continuously phagocytosis, and then it might lead to loss in the brain mass. And I may find that there are increase in plaque accumulation, as well as speed up the formation of tau, and the microglia shapes and size might increase and change compared to the parallel healthy model. I also have some concerns about the project, such as when we introduce the CRISPR edits to the mouse ApoE locus, we could unintentionally damage the genome as the locus is exposed to off-target cleavages. And moreover, these procedures might cause a double-stranded breaks, and then it could lead to other concerns about how to limit the structural variance, or we also have to consider like a low efficiency of these techniques, so that we have to screen through a lot of samples to find the design edit. And I also worried about some ethical issues when I'm working with animals models, is that the demand to minimize the amount of animals used, is that going to be effective to my projects or not? Or I need to consider about how and what proper housing and care conditions for the animals. And I also have to worry about the potential that I might cause more pain and suffering to the animal due to some invasive procedures. And then I also have to consider about the amount of pain relief that I'm gonna inject into these animals when I have to sack them, for example. And overall, this is my presentation about the concept map. Thank you for listening.